organs in the endocrine and reproductive system. Like hypertrophy, hyperplasia can also be physiological or pathological. An example of physiologic hyperplasia is enlargement of the female breast during pregnancy. In preparation for breastfeeding, hormones like prolactin, progesterone, and human placental lactogen stimulate the growth of glandular tissue in the breast and cause them to enlarge. An example of pathologic hyperplasia is having excessive hormonal stimulation. For example, normally during a menstrual cycle, the endometrium, which forms the inner lining of the uterus, grows and proliferates when exposed to estrogen. But if there's an overproduction of estrogen by an ovarian tumor, it can lead to excessive endometrial growth, also called endometrial hyperplasia. Now, one problem with hyperplasia is that it's sometimes associated with cancer. Normally, hyperplasia is an adaptive response to stress, so it's a tightly regulated process, meaning the tissue doesn't grow out of control. And in hyperplasia, the process stops if the stress factors are eliminated. That's different from cancer, where there's uncontrolled cell division. Now, the thing is that as cells divide in hyperplasia, some can mutate, and that's how hyperplasia can slip into dysplasia, and eventually into malignancy. While cells from hyperplasia are normal and healthy, cells from dysplasia can have an abnormal shape and function. And malignant cells might not only be abnormal, but also replicate uncontrollably and cause cancer. And that's why individuals with endometrial hyperplasia that's left untreated can develop endometrial cancer. Finally, it's important to remember that in tissues that have stem cells, hyperplasia and hypertrophy usually happen together when there's increased stress. For example, in pregnancy, the uterus gets stimulated by estrogen, which leads to hypertrophy and hyperplasia of the smooth muscle cells in the uterine wall. This means that the cells get tougher and the pack of cells gets more numerous. Alright, as a quick recap. Whenever an organ encounters any sort of physiological or pathological
tumor suppressor genes are involved in DNA repair mechanisms and inhibiting transcription. Oncogenes and tumor suppressor genes are classes of genes that code for various proteins that are involved in the progression of the cell cycle. Oncogenes are actually mutated versions of proto-oncogenes, which are normal genes in charge of positive regulation of the cell cycle. So, the protein products of proto-oncogenes stimulate cell growth and division. They're like a gas pedal in a car. Tumor suppressor genes, on the other hand, are in charge of negative regulation of the cell cycle. So, their protein products stop its progression and promote apoptosis, also known as cell death. Tumor suppressor genes are involved in DNA repair mechanisms and inhibiting transcription factors that try to push the cell along in the cell cycle. So, they're like the brake pedal in that car. Now, the cell cycle is the series of events that a cell goes through as it changes from being one cell into two daughter cells. The cell cycle has two phases, interphase and mitosis. Interphase is comprised of the G1 phase, during which the cell grows and performs its cell functions, the S phase, during which DNA is replicated, and the G2 phase, during which the cell grows again before entering mitosis. At the end of G1 and G2, there are cell cycle control points called the G1 and G2 checkpoints, where the cell checks to see if there's any DNA damage. The main control point is the G1 checkpoint. If it turns out that there is DNA damage, then the cell can either enter a non-dividing state called the G0 phase, where the DNA repair mechanisms try to fix the problem, or the cell can self-destruct in that process we talked about, apoptosis. But if the cell does get the go-ahead at the G1 checkpoint, it enters the S phase. And then, if the cell gets past the G2 checkpoint, it enters mitosis, and it divides into two identical daughter cells. However, once cells differentiate and become mature cells, like liver cells for example, they don't necessarily go through the cell cycle over and over again. Actually, cells tend to stay in that G0 phase, and some cells, like neurons, stay in G0 their entire life. These growth factors can be secreted by other cells, or by the cell itself, like when there's a tissue injury, and the remaining cells need to divide to replace the lost cell. Growth factors bind to growth factor receptors in the cell's membrane, which activates signal transduction proteins. Ultimately, that leads to increased transcription of genes that code for special proteins, like cyclins and cyclin-dependent kinases, so more of these proteins are being made. This is important because whether or not a cell gets cleared at G1 and G2 depends largely on the activity of cyclin-dependent kinases, which add phosphate groups to various proteins within the cell. And these cyclin-dependent kinases are, as you might guess, dependent on cyclin proteins. So what happens is that when there's DNA damage, the cell doesn't produce cyclins, and the cyclin-dependent kinases can't phosphorylate proteins within the cell, and that's the signal for the cell to halt the cell cycle. Okay, so proto-oncogenes code for proteins involved in promoting the progression of the cell cycle. Examples of proto-oncogenes include genes that code for growth factors or growth factor receptors, like the receptor tyrosine kinase or RTK, which adds phosphate groups to other proteins. Another example are genes that code for signal transduction proteins, like ROS genes that code for ROS proteins. ROS proteins are GTPases, meaning that they bind an intracellular GTP molecule and break it down into GDP and a free phosphate group. This further activates various cellular pathways, which ultimately result in cell growth, differentiation, and survival. Another example is the MYC proto-oncogene, which codes for a transcription factor that increases expression of cyclins and cyclin-dependent kinases. On the other hand, there are also proto-oncogenes that code for proteins that inhibit apoptosis. An example is BCL2, which prevents the activation of caspases, the enzymes that actually carry out apoptosis. Now, proto-oncogenes are normally only active when a cell needs to grow and divide, like how you only push the accelerator pedal in your car when you want to speed up. However, some genetic mutations like translocations, amplifications, or point mutations